Today we are going to be discussing transcription, translation, and single nucleic acid point mutations. To begin, we are given the DNA sequence shown on the screen, but before continuing, we must determine which strand is the template and coding strand. The template strand will be complementary to the RNA transcript, which is built in the 3' to 5' direction. This means that the bottom strand is the template strand. The coding strand should possess the same sequence as the mRNA transcript, although the thymine bases will be replaced by uracil in the mRNA transcript. This is the top strand in the DNA sequence shown on the screen. So the products of transcription will be an mRNA strand complementary to the template DNA strand. This is shown on the screen. Translation refers to translating the sequence of mRNA to a sequence of amino acids. From the derived mRNA sequence, the 4 amino acid long protein is tryptophan, isoleucine, threonine, and cysteine. Next, we are going to look at 4 different types of point mutations in the coding DNA strand and how these mutations affect the amino acids produced. The coding DNA strand with no mutations is shown at the bottom of the screen, with the location of mutations highlighted. The first mutation we will look at is a missense mutation, which causes a change in the amino acid produced. In this example, a thymine base is changed to a guanine base, so this mutation is a transversion. The original codon, AUU, coded for isoleucine, but the mutated codon, AGU, codes for serine. The second mutation is a nonsense mutation, which causes a codon to code for a stop codon rather than an amino acid. In this example, a cytosine base is changed to an adenine base, meaning that this is a transversion. The original codon, UGC, coded for cysteine, but the mutated codon, UGA, is a stop codon. The third mutation is a silent mutation, which is a mutation that still codes for the same amino acid. In this example, a thymine base is replaced with the cytosine base, which is a transition. The original codon, AUU, coded for isoleucine, and the mutated codon, AUC, still codes for isoleucine. The last mutation is a readthrough mutation in which a stop codon is changed to a codon that codes for an amino acid. In this example, an adenine base is changed to a guanine base, which is a transition. The original codon, UGA, is a stop codon, while the mutated codon, UGG, codes for the amino acid threonine. This concludes my exam number 6 mini video. Thank you for watching.